All right, this is an astrobiology presentation I did for my genetics class during my freshman year of college entitled Mitochondriony, the Origin and the Evolution of the Mitochondrion. This, of course, is the mitochondrion. It is the organelle which produces all of the chemical energy in the form of ATP, or adenosine triphosphate, necessary to fuel eukaryotic cells. So basically, all of your animals, all of your plants, all of your fungi, they need mitochondria to survive, and it seems to be necessary for what we call complex multicellular life. So complex multicellularity is what we see in animals and fungi and plants, and we don't really see it in prokaryotes. Although there are forms of what you could call multicellularity among prokaryotes, they never organize into big, complicated macroorganisms like animals and plants and fungi. We really only see big, complicated life forms like that among the eukaryotes. So how did eukaryotes first arise? Well, over here on the right, there is a family tree indicating the genetic relationships of various prokaryotes to the eukaryota or eukarya, which is eukaryotes like us, with our closest relatives being various members of the archaea. So the Euryarchaeota, the Crenarchaeota, the Thaumarchaeota, the Eigarchaeota, and the Chorarchaeota, these lower four of which are together considered the TAC group of the archaea. There are other forms of archaea too, but they aren't shown here. And there's actually one group of archaea that has shown to be even more closely related to us, the eukaryotes, than any of these guys here on this list. But they're not indicated because where I got this image from was actually slightly older. The discovery of this new form of archaea, they're called the Asgard archaeota. That's actually fairly recent. I don't think they were discovered until the last 10 years or so. But that's not really vital to this particular presentation. So anyway, we're just going to get on with it. When we look over here on the left, this shows basically the timing of when eukaryotes became recognizably eukaryotic relative to the mitochondrial acquisition. This top hypothesis here, or well, category of hypotheses, we'll call the Archeozoan hypothesis, or Archeozoan hypotheses. And they basically state that a relative of the archaea branched off. These are the archaea here. A relative of them branched off and became more or less eukaryotic. They had a nuclear membrane and looked more or less like eukaryotic cells, except they didn't have mitochondria yet. And one of them at some point acquired a mitochondrion and became modern eukaryotes. Eukaryotes as we know them today, who have mitochondria in their cells. Now this bottom hypothesis, or rather set of hypotheses, are what we would call the symbiotic hypotheses, or symbiogenic hypothesis, which basically states that a member of the archaea, and this little branch here indicates a member of the archaea. All of these guys are basically archaea, but a member of the archaea here acquired a mitochondrion here, and the mitochondrial acquisition, according to this hypothesis, is what led to eukaryotes developing most of the features that we associate with eukaryotes today, like, for example, a membrane-bound nucleus. And if you're wondering what any of these acronyms mean, LUCA here, L-U-C-A, that stands for Last Universal Common Ancestor. So that would be the last common ancestor of all living things on Earth today, or at least all cellular life forms. So all of what we call the cytota. And LECA, L-E-C-A, indicated here, that stands for Last Eukaryotic Common Ancestor. And if you're wondering what this B stands for, here, that's the bacteria. In case anybody is wondering, the mitochondrion in our cells appears to be genetically most closely related to a group of the proteobacteria known as the Rickettsiella, which is the group of bacteria that includes the pathogens who cause rickets, and that appears to be the ancestor of the mitochondria. Meanwhile, the nucleus of our cells, the DNA, our nuclear DNA, the DNA in the nucleus of our cells, that appears to be more closely related to the archaea. So really, eukaryotic cells are composite cells, or if you'd prefer the terms Frankenstein cells or Cronenberg cells, they're basically combinations of bacterial and archaeal cells. Now, of course, there are a number of hypotheses for how this might have happened. Over here on the right, we have what I just referred to as the Archaeozoan hypothesis, the idea that an already more or less eukaryotic cell evolved and then acquired the mitochondrial endosymbiont. So we start out with a prokaryotic cell, which was an archaeon, we know that now, and it had what we call a nucleoid region, which is a region in the center of the cytoplasm that contains the genetic material. And in this scenario, the outermost membrane of the eukaryotic ancestor, its membrane started to fold in on itself and wrap the nucleoid region in sort of a secondary membrane, which became the nucleus, and then, of course, acquired a proteobacterial endosymbiont, which became the mitochondria. And this particular image also shows another step that occurred in plants where cyanobacteria or blue-green algae endosymbionts were acquired, which became chloroplasts. But that's not really relevant to what we're talking about now. What we're really talking about focuses mainly on this part of the image here, which is the evolution of an archaeozoan or a proto-eukaryote prior to the mitochondrial acquisition. Now, that is in contrast to what we have over here, which is the symbiogenesis first hypothesis, which basically has an archaeon with a nucleoid region acquiring an alpha proteobacterium, and that triggers the changes which cause the nucleus to evolve, possibly to separate the DNA of the mitochondrion with the nuclear DNA that codes for the features of the organism itself. Below that here, we have the serial symbiogenesis hypothesis, and that's meant to explain the fact that the outermost membrane of eukaryotes really seems to have more in common with bacteria than with archaea, even though the nucleus of our cells, the main DNA that codes for all of our features, that is clearly more closely related to the archaea. But the outermost membrane seems to be more similar in many ways to bacteria. So with the serial symbiogenesis idea, we have bacteria engulfing an archaeal cell, And that archaeal cell, the whole thing becomes the nucleus. Instead of the nucleoid region becoming the nucleus, the whole archaeal cell becomes the nucleus. And then it acquires an alpha proteobacterium and becomes the first eukaryote. And of course, down on the bottom is another hypothesis, which basically has the alpha proteobacterium and the eukaryotic ancestral archaeon having an already existing symbiotic relationship, and then the archaeon extends its cytoplasmic matrix outward, especially the cytoskeleton, to sort of protect or house the alpha proteobacterium, and that's how it became the eukaryote. So, oh yeah, and I should mention that this version up here, the serial symbiosis hypothesis, it really doesn't have a preference as far as which came first, whether the outer membrane of the eukaryotic cell was acquired from bacteria before the mitochondrion, or whether the mitochondrion came first. So like these two, you could kind of switch them around and it doesn't really make that much difference. Those are both hypotheses. There are literally all kinds of eukaryogenesis hypotheses, and this is just a brief overview. If I were to go in depth, I would be talking for hours. So I'm kind of skimming over most of this stuff. Another hypothesis for why we eukarya seem to have such a hodgepodge of features, why our nuclear DNA seems to be so much more similar to the archaea, and why we indeed seem to be a member of the archaea, and why our outer membrane seems to be more similar to the bacteria, is that our common ancestor, the LUCA, the common ancestor of all living things, might have existed before the evolution of cells, before there was an outer membrane, actually, prior to what Carl Woese called the Darwinian threshold. Now, if you don't know who Carl Woese was, he is the person who came up with the RNA world hypothesis, and he was the first one to notice that the archaea were not a type of bacteria, as had been previously believed. The archaea used to be called the archaebacteria. That term has since fallen into disfavor. There are only a small number of scientists who still use the term, and it's considered something of an anachronism or an archaism. It's not really considered entirely appropriate to use the term archaebacteria anymore. We call them the archaea now because they are decidedly not bacteria. Carl Woese was the first one who noticed that the archaea seemed to be more closely related to the eukarya than to the bacteria. Even though superficially they resemble bacteria, they don't appear to be nearly as closely related to the bacteria as they are to us. And he hypothesized that there should actually be 3 domains of life instead of prokarya and eukarya. It should actually be bacteria, archaea, and eukarya. And that the archaea and the eukarya are sister groups together in a clade called the archaea. And basically, the 3 domains of life did not fully diverge until passing this Darwinian threshold. Before that, we just had a bunch of chromosomes floating around in the RNA world, virus-like replicons going in and out of little protocells that you could call progenotes. And there were circular chromosomes indicated here. There were linear chromosomes like we find in our nuclear DNA. Circular chromosomes are what we find in prokaryotes and in our mitochondria. And basically, the LUCA was a pre-cellular life form, and actual cells began to evolve much later. and they had sort of almost a random assortment of features. So we end up with archaeal-like DNA and bacteria-like membranes, and then we get mitochondrial acquisition, which leads to the eukaryotes. And so basically, the idea here is that the Luca did not have cells yet. That actual cells um, evolved much later. Now, of course, Carl Woese did not agree with the symbiogenesis hypothesis for mitochondrial acquisition. He kind of believed, like a lot of people prior to him, that the mitochondrion broke off from the nucleus. Um, so what's shown here in this image to the right is actually Eugene Koonen's modification of Carl Woese's precellular Luca hypothesis, which reasserts the endosymbiosis hypothesis for the origin of the mitochondria, which Carl Woese did not accept. That was one of his major downfalls. Even though he contributed a lot to the study of the origin of life and to the study of eukaryogenesis, one of his major failings was he was never willing to accept endosymbiotic theory. So over here we have a slight modification of this idea according to Eugene Koonin. Now, there are two main approaches to the origin of life problem: the replicator first approach and the metabolism first approach. And these can be taken either literally or figuratively. For example, in the replicator first approach, that can be taken literally to mean that self-replication literally predates metabolism, or it can simply mean that you're putting greater emphasis on the origin of replication when it comes to the origin of life. That self-replication is the most important thing to consider when talking about the origin of life. It doesn't necessarily have to mean that self-replication literally came first. It can mean that, but it doesn't have to. So that term can be either literal or figurative. And the same thing with the metabolism first approach. That can literally mean that catabolism and anabolism predated self-replication, or it can be used more figuratively to say that the origin of metabolism is really the most important thing to consider when talking about the origin of life. So it can refer either literally to when these events actually occurred, or it can refer to where you prefer to put your emphasis and what you think is really the critical step to what we call life. Now, with the replicator first approach, you'll see here this references the RNA world hypothesis, which I just spoke of, which was first hypothesized by Carl Woese, and that has to do with the origin of self-replicating bits of RNA. And the metabolism first approach basically has to do with the coupled reactions that can occur inside a cell. The metabolism first idea mainly has to do with the lipid world hypothesis, and where the first lipid bilayers came from that form the cells that RNA would later come to inhabit. And one really good explanation for this sort of chemical evolution, or the lipid world hypothesis, is hydrothermal vents. Anybody who's read about the origin of life or has seen a blurb of it in a biology textbook has probably seen this hydrothermal vent hypothesis. And that works really good for explaining where a lot of the chemicals inside our cells might have come from, and of course where the cells themselves might have come from. But it's really poor at explaining where RNA would have come from. RNA really likes to break down pretty easily. A lot of the nucleotides like adenosine, cytosine, guanine, uracil, they don't really last very long at temperatures much above room temperature. It's better in DNA when you have like a sugar phosphate backbone, or in a fully formed RNA strand where you have a sugar phosphate backbone. But as far as like trying to get them to form to begin with, um, they break down really, really easily at warm temperatures. And so um, the RNA world hypothesis really works better if you assume that life started on the surface of a planet, or at near freezing temperatures, whereas metabolism first approaches those seem to do better with the hotter temperatures and hydrothermal vent type hypotheses. And of course, no origin of life discussion would be complete without mentioning panspermia. Although technically panspermia doesn't necessarily have to do with how life began, it does have to do with how life came to this planet, or more generally, how life ends up on planets other than where it first started. Here we have what's called lithopanspermia, which is basically transport of life forms via rocks across space. This picture here shows what's called Earth-Mars interplanetary lithopanspermia, which is basically the idea that like a meteor or something hit Mars, a bunch of impact ejecta was kicked up, and some life forms that might have been in the rock that was kicked up end up on Earth. Of course, many famous scientists like Neil deGrasse Tyson, for example, have uh, have really championed the idea that Mars was wet and fertile for life before Earth was, and so that's kind of seen as a very plausible scenario for how life made it to Earth, although it completely dodges the question of where life originated to begin with. Um, which panspermia isn't really meant to answer. Although when we get to molecular panspermia, the other form of panspermia, that really has more to do with the kinds of chemical reactions that could lead living things to evolving to begin with. So, for example, we can have chemicals forming in giant interstellar gas clouds, like the one that the pre-solar nebula formed from. And of course, pre-solar nebula become protoplanetary disks, and planets form out of those, and life might evolve on those planets, and then meteors strike the planets and blow the material back up into space, and then that material can end up back in interstellar gas clouds again, and it can become a sort of a sort of feedback loop. And so it's possible that our galaxy went through many generations of this occurring before what we call true life forms could have evolved. Now, one of the main reasons why we consider interplanetary lithopanspermia, for example, Earth-Mars interplanetary lithopanspermia, to be so plausible is because numerous microbial life forms are really good at surviving just insanely extreme conditions. For example, there are numerous species of bacteria who are good at surviving low pressure, low temperature, low moisture, high radioactivity, even ionizing UV radioactivity like we find in space. There are also numerous bacteria whose spores can survive high acceleration and high impact. Everything you would need to launch these little critters off the face of a planet, have them survive a trip through space, and land alive on another planet. But we didn't evolve from the bacteria. If you remember, the mitochondria in our cells evolved from bacteria, but our nuclear DNA, our DNA, the DNA that codes for um, our life form, our phenotype, um, that evolved from the archaea, not bacteria. And when we look at the archaea, we see something completely different. The archaea, who are famous for being extremophiles, are especially famous for being able to survive high pressure, high temperature, high acidity, high salinity, high alkalinity, decidedly planetary extremes. Now, there are exceptions, of course. The most radiotolerant of all organisms isn't a bacterium, it's an archaean. Uh, uh, Thermococcus gametolerans, for example, is the most radiotolerant organism. So there are exceptions all over the place, but in general, it really seems like the bacteria are better at surviving space-like extremes, and the archaea are better at surviving planetary extremes. So there appears to be a bit of a contradiction here, because even though many species of microbial life forms are good at surviving really crazy extremes, the microbial life forms that we evolved directly from seem to be much better at surviving decidedly planetary extremes. The microbial life forms who are more famous for being able to survive space-like extremes are not the ones that we evolved directly from. They're the ones that the mitochondria in our cells evolved from. So how do we harmonize this? Do we rule out panspermia as a possible origin for life on Earth, or do we say, well, maybe the archaea, our ancestors, evolved on Earth, while the ancestors of our mitochondria, the bacteria, um, evolved on another planet and came here later on? Um, well, that's kind of hard to swallow because there are over 300 genes that are shared by all life forms on Earth, including the bacteria and the archaea. So ultimately, we have to share a common ancestor. So how would this work? And that brings us back to molecular panspermia. When we look out in the universe, we find that most of the elements that are essential for life, for example, carbon, oxygen, hydrogen, nitrogen, those are found all over the universe. Those are some of the most common elements. And we found potassium and phosphorus in comets and in meteorites, and we found them in the remnants of supernovas. We found amino acids in meteorites, for example, the Murchison meteorite. Also in the Murchison meteorite, we found the purines adenine and guanine, which we need to make RNA and DNA. We found the pyrimidine uracil, which is in RNA. And we've also detected cytosine in the Murchison meteorite, but not at high enough above background levels to confirm that it came from the meteorite itself. Although experiments that attempt to replicate astrobiological conditions or astrophysical conditions, we have managed to produce cytosine in such experiments, and we've also managed to produce ribose in such conditions. I've sort of marked those with a star to indicate that we haven't directly detected those in space, but we have produced them in experiments replicating space-like conditions. Although ribose has, since the time that I first made this presentation, been found in meteorites. So that can be considered kind of a confirmation of the hypothesis. This one, there shouldn't really be a star on it anymore because we have found ribose in the Murchison meteorite. And meteorites like the Murchison meteorite are formed from some of the oldest material in the solar system. Carbonaceous chondrites probably ultimately come from before the formation of the planets, from the protoplanetary disk. Um, so how do we harmonize this? How do we bring all this together? Well, what if there were two completely independent metabolism first lipid world type scenarios going on, one on Earth, one on Mars, but they both received genetic material from another source, like the protoplanetary disk, for example? What if RNA formed in the pre-solar nebula or the protoplanetary disk, and then ended up on Earth and Mars, where the lipid world type scenario was going on, landed on two different lipid worlds and evolved into two completely different types of life forms? archaea on Earth, and bacteria on Mars. That could explain why we're so different, why the bacteria seem so good at surviving space-like extremes, because the bacteria we have on this planet may have anciently survived a trip through space. Meanwhile, our direct ancestors, the archaea, seem to be much better at surviving planetary extremes because they're actually native to this planet. And of course, ultimately, we do have to share a genetic ancestor, but that genetic ancestor might predate living things. It may not have been a true living thing yet. So, in this scenario, we could have two origins of life, or two origins of true cellular life, but with one common source of genetic material. That could explain how we could come from two different planets and still share a common ancestor. Anyway, the paper that went along with this presentation can be found at genesispanthesis.org/mgeny, short for mitochondriogeny, m-geny, geny.html, and that is at the Genesis Panthesis website. So please go give it a look. And that's the end of the presentation. So thank you very much and have a great day.